Hello, everyone, and welcome to WorkCare's briefing on preventing and managing COVID-19 in the workplace. The webinar will start in just a couple minutes.
Hello, everyone. Thank you for joining us for week 18 of our briefing series on preventing and managing COVID-19 in the workplace. This is Karen O'Hara, Director of Marketing and Communications for WorkCare. I'm your moderator. You are on mute. Please submit your questions using the question and answer function on Zoom. The webinar lasts about 40 minutes and it's being recorded. We always post a recording on our YouTube channel, and Dr. Harris summarizes answers to all the questions which we post on our website every week. It's really a great resource if you consider we've been doing it now for 17 and just getting ready to do number 18. So I encourage you to look back for any questions you may have. It's a great record. As you all know, if you've joined us before, or if you're just joining us for the first time, I'd like to introduce our weekly speaker, Anthony Harris, MD, MBA, MPH. Dr. Harris is our Chief Innovation Officer and Associate Medical Director for On-Site Clinical Operations. He's also our lead clinician for our COVID-19 response. I'll let him jump right in, so save time, and we'll take as many questions as we possibly can in the 40 minutes that we have allotted for the briefing. Thanks again for joining us.
Excellent. Thank you, Karen, and thanks everyone for joining us once again. Happy to be with you for week 18. And this week, we're going to take a step back. And what I mean is that we're going to examine some of the concepts, some of the things we've presented in the past, and we're going to give general updates to that. Amongst these items are, the trend that we're on right now in terms of the increasing cases that we're all obviously aware of, and just getting general updates on what does the future look like for us, right, on a number of levels. And so we'll jump right into it here. The process and the standard approach will be the same. We'll do a clinical update, followed by some pathology, microbiology updates here, and then review what is the trend from an epidemiology standpoint, talk about some prevention, and finally, the workforce strategies will be kind of an update again about what's relevant, really focusing on what's relevant for us in the workplace, and kind of what things look like again prospectively, and what to expect in the summer and the rest of the summer and the fall. So without further ado, jumping into the clinical update. Not much new in terms of the clinical symptoms, but obviously we're seeing this anecdote here in terms of supernova. This is actually a NASA capturing for the first time visible spectrum of a supernova as it explodes. So this is a gift, but it is an actual supernova that occurred. So I know it's a little jocular here, but in all seriousness, we're seeing an explosion of these presentations, and it's coming at a time where obviously we're starting to get into that flu season. And so we're seeing individuals at a increasing rate being kept home because of presentation of symptoms that may be SARS-CoV-2 related, COVID-19 related, or may not be. And so therein lies the challenge, right? How are we going to effectively differentiate who has a cold or the flu or who has COVID, and the relevancy of that being who needs to stay home for 3 days until their symptoms improve versus who needs to stay home for 10 to 14 days until their transmission potential of COVID-19 has expired, that window has expired. So we'll be talking more in depth about this as we move forward here in this presentation. And so if we look at then kind of giving an update from a pathology standpoint, we oftentimes will update on vaccines, and that's what we'll do today. So if we look at what the vaccine landscape looks like, if you have not heard of this, it's been around, we'll update on it. It's called Operation Warp Speed, OWS. I know it sounds all kind of futuristic, if you would, but it kicked off from the federal government back earlier in the year, back in March. And in April, funding was authorized by Congress to help speed along vaccination of the general public here in the US, right? And so what is it? We won't go through the entire document. It's available for your review at the link here presented. But in short, they kicked this off with major funding with 3 partners, one being Johnson & Johnson back in March, $456 million in funding. The second was Moderna, and they received $483 million in support, and that was back in April. And then in May, $1.2 billion to AstraZeneca to help develop a vaccine in conjunction with University of Oxford, okay? And so these were the main 3 selected candidates that received federal funding to go after bringing a vaccine to market more quickly than we have been able to do historically, right? We've touched on that before in the past in regards to the toll gate for FDA approval of a vaccine between preclinical phase 1, phase 2, and phase 3 trials, and that period taking years, in some cases up to 10 to 15 years, and obviously accelerated here with some substantial funding. And there are organizations within the federal government in conjunction and falling under the secretary, one of the secretaries of health under HHS, but it's a committee and authority called Biomedical Advanced Research and Development. And so this is the organization that spearheaded a lot of this funding, a lot of this collaboration with private institutions. And we want to continue to pay attention to these efforts that are being made, obviously, one, because we're interested in when a vaccine is going to be available to the general public, and two, it's good to know that there are measures being taken to really bolster the capabilities of both public and private institutions. And so to update on a July update here with a congressional testimony recently with National Institutes of Health, we want to update on, we won't update on every single, obviously, effort. We don't have time for that in this context, but we do want to shine some light on what the potential is to have a vaccine later this year or early next year. And so this testimony gives us some indication to that. This testimony authored here that the collaboration with Moderna in particular published some interim findings after phase 1 back in May, late May, okay? And they kicked off phase 2 trials as well. And those studies have reported at this point, and now they're considering launching the phase 3 clinical trials this month as well. So obviously good news there in that this has been able to progress through the development phases towards approval, gives us hope, right? If we look at then further in the testimony, that collaboration we highlighted with Oxford that received the $1.2 billion, they studied the vaccine, or they derived the vaccine from a chimpanzee adenovirus vector. We'll touch on that a little bit here, but they've gone into phase 3 and been in phase 3 for some time in the UK, right? Multiple locations in the United Kingdom. And so that's good news. And if we examine that further, we presented this maybe about 2 months ago, and then updated about a month ago. Today, we'll provide another update because we're not seeing additional phase 3 trials at this point, but we are seeing progression of these phase 3 trials, meaning that they are expanding their efforts. And again, Oxford being one of the darlings, if you would, in terms of hope for a vaccine to be brought to fruition later this year. And so they actually started launching, as of this month, trials beyond the UK and Brazil in this case, and looking at launching of trials in other locations. And so again, in this particular launch, I believe 3,000 participants, volunteers in Brazil were identified. So in short, we'll touch on this just to show you how the Oxford study differs and how they derive the vaccine from some of the other efforts. In short, what they did was took an adenovirus, a virus that can't cause disease in humans. They then spliced with that adenovirus the spike protein. We touched on that spike protein, the S protein, before in previous presentations, but that's the protein that we know has mutated in some studies up to 14 times at this point, right? But nonetheless, that's the important protein in many of the vaccines to be identified by the host, us, of being foreign. And that's what our immune cells will attack. And so in this case, they place that protein into the adenovirus, and that's what makes up the vaccine. We are vaccinated with that. Our body recognizes that, in this case, the vaccine that can't cause us any harm from that adenovirus from a chimpanzee that has the spike protein. And then if we should be exposed to SARS-CoV-2, our body is able to also identify that as foreign and quickly mount an immune response to render that virus ineffective in terms of its multiplication in our system. Okay, so that's how this particular study, again, one of the most hopeful studies in phase 3 clinical trials, looking to do manufacturing as well. New partnerships were announced here in the US. And they are anticipating, if the trials continue to go well, bringing to fruition a completed vaccine in as soon as October of this year, or at the early onset of next year, 2021. All right, so that's the quick update in regards to vaccines on the pathology front, microbiology front. So let's look at the epidemiology. We'll kick it off as we normally do, looking at the numbers in the US. Starting 2 weeks ago, where were we? 2.5, almost 2.6 million cases. Then we moved to 2.9 last week. This week we jumped up another 400,000 almost. So 3.3 million cases in the US at this point. And we're up to 135,000 deaths. Hold that number in your mind. We'll talk about that number and the projections that we'll present here in a couple minutes. But 135,000 deaths currently in the US. We can see the states that have the most burden of disease here as well. Now let's look at this map, a little different here. We haven't presented this map before from the CDC, but it's just looking at the deaths now, not just the incidence or prevalence of the illness in this map. We are going to look at the deaths and the states that are most affected by 5,000 or more deaths are highlighted in that dark blue there. So we'll continue to update with this particular map in coming weeks here. If we look at then cases from the CDC published, new cases day by day, we want to watch this trend. This was last week. And this is this week. The trend continues to grow, right? And so we're seeing worse cases than we've seen yet in the presentation of COVID in the US. And so we want to watch this and then compare it, how we're doing to the rest of the world. And if we look at that from 2 weeks ago, we saw the growing trend, right, scaling up of our total cases in the US with this 7-day rolling average. And then we continued that trend last week. And this week, obviously, we're continuing that trend still. But we're looking at this a little differently. This is a new metric here in terms of not just looking at the daily new confirmed cases in the 7-day rolling average, but now we have layered into this a relative map of or understanding, depiction, if you would, of how we're doing compared to other countries in regards to that number we've been watching, positive rates of tests being conducted, okay? And so if you see countries in the dark blue here, they're trending down and they are in a range where their testing is adequate, most likely, to articulate the burden of disease in their particular country, right? Because they've scaled their testing enough to be in the range where it's likely that they have an accurate picture. If you look at the US, we're in that 3 to 10% range. We've been in that range for a number of weeks now, but still we're not yet blue, meaning that we have work to continue to do to scale our testing capabilities here in the US to get down into that 2 to 3 or 1 to 2% range as other countries have done. And so this also plays out in terms of how these countries have fared with regard to their burden of disease, right? The better they've been able to scale their testing, the better they've been able to respond appropriately to COVID. And that has also played out positively in a significant way. So we'll continue to watch this particular figure. If we look at then the deaths as it correlates to the bump that we've seen in terms of number of cases, we saw this 2 weeks ago with that sharp increase. Then we saw a decrease here in the overall 7-day rolling average here. And then we're seeing another little bump, okay? So we'll, again, continue to watch this. We know that and we'll talk about the projections for deaths as we continue to move through the summer and into the fall. And we'll get a little bit more granular here in just a moment. So now let's talk about updating the preventative programs that we've looked at in the past and getting a good update, okay? So we've looked at testing. Testing, obviously, at this point should be a part of your process, your protocol of prevention, but there are still issues with testing, okay? And so we're gonna get a little deeper into what the problem is, articulate it in such a way that hopefully we'll watch it over time improve, okay? So if we look at the myriad of tests that are available in the US right now, there's quite a bit, right? And this is a list coming out of Harvard examining the performance and comparison of different types of tests. But far and away, the #1 test you're going to encounter in the marketplace is PCR tests. And we're talking germanely about those tests that have been FDA EUA. That's the emergency use authorization, okay? And so these are the ones that, for the most part, have some level of reliability in regards to they've been vetted by the FDA and authorized authorized for us to use, and we have some benefits around that from a medical legal standpoint, as well as employers do as well when we're using these particular types of tests. But we want to see that in terms of these specific PCR tests, the issue that still remains, and the issue comes in around sensitivity and specificity. Okay, we've touched on these particular concepts in the past. We will refresh that briefly here. Sensitivity is how likely am I to detect illness in somebody when I perform this test, and specificity is how likely am I to diagnose that a positive result is the disease I'm looking for, in this case COVID-19 or SARS-CoV-2. Okay, and so the higher your sensitivity specificity, the more accurate the test. The issue lies in here is how can we clinically call somebody or consider somebody to be not positive or negative for COVID-19? Meaning, if I do a test, is that test in vain, or is that test actually going to help me make a clinical decision on my worker that I'm testing? All right, and so we won't jump into the details on pre-test probability and post-test probability. That's a whole lecture in and of itself. But what we want to examine in particular is a depiction of if the sensitivity, so if we hold the specificity the same as we are in both of these curves here, if we look at just the sensitivity, how likely am I to detect the illness I'm looking for? 70% sensitivity is where we started when PCR first came onto the scene. Many of the tests were around that range of accuracy. We only were able to, in the order of 15% less than a higher sensitivity, call somebody negative for COVID-19. Whereas if we have a sensitivity of 90%, then we're far more able, from a clinical standpoint, 15% better accuracy in terms of considering somebody from a threshold standpoint negative for COVID-19 when they receive a negative test. All right, and so we want to make sure we're using tests that have that high sensitivity and specificity. Now, here's the crux of it, right? If we look at the number of tests, as we have in the past, of FDA, EUA approved tests, PCR and other, in this particular case, we're looking at and isolating those nucleic acid tests, the PCR amplified tests, right? We know that there's over 100 of these available right now. But what we want to examine in detail is, of these tests that have been authorized by the FDA, do we have data on their sensitivity and specificity? Those are the two columns here that are in the right portion of the highlighted region, okay? Where you see those 100%, you're also seeing some blanks. And those blanks are meaning that the data is not available. And this, again, coming out of published on Harvard's website here, and data they've been able to find. Anecdotally, and within work care, we've not been able to find a lot of data on tests that have been available. For example, the Abbott ID Now test, still no data available widely on the sensitivity and specificity. And in practice, we all know that that test had a lot of recalls because of poor performance, right? So therein lies the crux. Your worker gets tested at a local clinic, at a local lab, and we don't have data. You may or may not have data on the accuracy of that particular test being performed. So as we ramp up access to testing in the US, we also need to be very cognizant of, do we have data on the tests that are being performed? Obviously, this is more germane to testing programs that you're instituting with your clinical partners, right? If you're sending people to a local testing facility, obviously that may be the choice of the employee as to where they go. But if you as an employer are putting in place a program, you want to make sure you are validating the accuracy of these tests because it's data that oftentimes, unfortunately, it's hard to get. And if we look at the column here in isolation, we can see the paucity of data that's available for all these tests. And that's just page one. Page two is very similar. Some of these tests that are available data have great numbers, right? But that is also telling because the published data usually is good data. The ones that are missing data don't have the data published because the reliability and accuracy is probably not so good, right? So just be mindful. This data is public, so just be mindful to ask these questions as you're developing these programs appropriately, so that you're making that clinical decision making possible. And how that plays out, right, in terms of questions that you're trying to answer in the workplace, it starts with, hey, can my worker come back to work? Do I have to do contact tracing for this worker? Was this a true exposure? Does the worker need self-quarantine, etc., right? These questions become difficult to answer if you don't have the accuracy data of the tests that were performed on these workers. So again, pay attention, be mindful to ask these questions, and put in place the right test for the right program so that we're effective at our preventative efforts. And so what does this all mean at the end of the day? It means our clinical protocol, even with some tests in place, if we can't validate the accuracy, haven't changed. That people who are either presumptively positive, workers who are presumptively positive, workers who are diagnosed positive from laboratory, still may have to do a full 14-day quarantine, because we can't get them back to work more quickly, right, without high accuracy in the tests that are coming back as negative, right? So again, it does impact, at the end of the day, our ability to be effective at preventative strategies, both primary, secondary, and tertiary. And so just a reminder, find the accuracy data. If you can't, think about it, think twice before instituting that into a preventative program. So as we look at then kind of a recap of strategy, and as we recap and talk about then prospectively what the future may hold for us, right? We want to touch on some key elements that we've very early on touched on, right? And that was the strong social distancing measures that needed to be in place to stem the tide of a major outbreak, a pandemic scenario, right? This we presented, I believe, on one of the first, within the first weeks of doing this presentation. And we saw that, you know, if we do have in place a very strong system for social distancing, we can achieve limitations to the outbreak. We looked at this Stanford study also talking about the duration of strong social distancing and how long something should be in place in order to be effective. And we looked at it from a 3-month, Stanford looked at it from a 3-month perspective, how the curve was bent at 5 months, right? We're talking into October at that point, 2020, and then at 10 months. And at 10 months, yes, we saw obviously a dramatic decrease in the transmission of COVID-19, right? So obviously that didn't happen. This is the current map of states as well as counties, and it's color-coded based upon, is that county experiencing in the red more cases, increasing incidence per 100,000, or are they experiencing a decline, fewer cases per 10,000, excuse me, not 100,000, but per 10,000 in that particular county? If it's in that gray kind of whitish zone, that means they're kind of steady state. They're not really experiencing too far to the left or to the right changes in the incidence, okay? And so we want to pay attention to this because it's important to help us, again, understand what our local response should be, right? But if we look at some of the correlations to this data, on the top, you see amongst some of the states that are suffering right now, that are experiencing some of the highest burden of illness. We presented this data some time ago in regards to the shaded area on the top and the bottom, and that shaded area representing the duration, the total duration of that state's social distancing orders, okay? And so obviously we're seeing, and so on the bottom, we're seeing to the contrary, the states that are seeing dramatic decreases in the number of cases over time, and their shaded area and the total duration of their strong social distancing orders, right? And this, you know, retrospectively shows us that what we anticipated in terms of strong social distancing and the effects on transmission have played out as we predicted, right? And so again, this gives us insight as to how we can understand what the future is going to look like in regards to, is it going to continue to get worse, or is it going to hopefully get better? And so if we want to go deeper into those projections, obviously we don't have to do it de novo on our own. We can look to universities and data published by the CDC for these projections. And so on a weekly basis, the CDC is receiving a number of forecasts from different universities and institutions. In this case, 23 individual national forecasts were performed and delivered to the CDC, and they put this data together so that we can look at it in summary format here. And if we look at the first chart here on the left-hand side with the national forecast, we're looking at time as it approaches August 1st on the x-axis, and on the y-axis, the cumulative reported deaths, right? And so again, we know that the deaths do correlate to the number of cases. So this gives us a model for understanding the level of cases that will play out as well. And so if we look at this data, we can see at the top range, about 175,000 deaths we're going to reach estimated in these projections by August 1st. It's between 140 and 160 in general. But again, if we look at these numbers in aggregate and we take it as a mean, then we're about 150,000. So remember the number I asked you to hold in your mind previously with regard to where we were with the US numbers as it stands today, right? That number was 135,000 deaths. So by August 1st, that number is going to swell, estimated to swell to 150,000, even higher, maybe 160,000 deaths by August 1st. And so obviously that's a dramatic increase from where we were. We were increasing at 126 2 weeks ago to 130, 4,000 cases, to 135, 5,000 cases. Obviously to get to 160, we got to increase by another 25,000 cases, right? And so again, that is a still dramatic increase within the next couple weeks here at the end of July. And that gives us an idea of what the rest of this summer and the fall is going to mean. This is not even taking into account, right, the return of students to the schools as it might play out in many states. So not to be too dramatic in the prognostication of what's going to happen, but anticipate things to get worse, right? And we can see this play out state by state and available data from the CDC. So if you're interested in that, looking at your particular state or your jurisdictions, please go to the CDC website to see these projections and make sure you have the right game plan in place for the impact that may come about. And so what do? I mean, this is pretty grim in terms of our prospect of COVID-19 in the fall, right? But the kind of the go-to is to stay the course, right? Make sure you have your plan in place for your tertiary, secondary prevention. Make sure you're doing the things that in this case, the game plan that the feds laid out long ago said employers should be doing: the pre-screening, the regular monitoring, obviously mandating masks in place. Make sure you have the different components of the preventative strategy, right? The screening, the COVID surveillance program. Make sure you're prioritizing your geographic risk appropriately for resource allocation. And obviously a testing regimen needs to be a part of your process, right? If you're doing these things, then you're going to be able to limit the amount of transmission in the workplace. Now, obviously, as employers, there's a limit to the impact you can have for community transmission, right? That takes a higher level of organization at this local, county, state level, et cetera, national level. But within the workplace, even though we may see in the workplace, and we are anecdotally, increasing number of people being kept home, increasing number of positive cases, that trend that we're seeing play out in the workplace, it may in some cases be very positive that we are identifying early those individuals at risk or becoming positive and getting them home safely. What may be more ominous in regards to not having the right program in place is that you're seeing a local increase in incidents but in the workplace, you're not seeing a trend that mirrors that, right? That means you're probably missing many positive cases or those individuals at risk in the workplace. So go back, look at your KPIs, look at your metrics, and see and make sure that what you're seeing in the workplace is playing out in your local, is matching what you're seeing locally. And if not, if you're underestimating the amount of illness in the workplace, you might not have the right program in place to do that primary preventative prognostication, if you would, of keeping people home safely. So with that being said, that was a good recap here of kind of where we were, where we're at, where we're heading, and hopefully we have some good questions to answer. Thank you.
Thank you, Dr. Harris. As usual, we have quite a few questions to get through, so I'll jump right in. Several people are asking about the veracity of CDC data, and I know this is partly a political question, and you don't typically like to answer political questions, but there's been a lot in the media about the CDC's ability to collect data, the accuracy of the data, and then one person asked, what are the implications of the government deciding to prevent hospitals from reporting COVID data to the CDC, which is something I hadn't heard about. Could you comment on it in general? And then I know you've cited a number of other sources for data that you're relying on also, but maybe you could elaborate.
Absolutely. So yeah, a multifactorial approach to understanding the data is definitely a must, right, at this point in time. And we strongly recommend not having a sole source of data being the CDC. Now, with that being said, the CDC does serve as a springboard to look for other sources of data. And, you know, at the end of the day, there is a lot of good data that the CDC captures, but then be diligent in going back and looking at specifically where that data is coming from. And a lot of institutions, obviously a lot of non-for-profits have data capture going on, and it's not just US based, it's international, and looking to those as sources for understanding trends is definitely important. And obviously any attempt by anyone at any level to suppress data obviously is going to do a disservice to all of us. So my hope is that the efforts to gather more data are going to be supported at all levels versus the opposite.
Okay, a couple questions about testing. In the test-based strategy in a return to work protocol, if a saliva test is performed, are 2 negative tests still required, or is one sufficient based on accuracy?
Sure, it does come down to, in particular with the saliva test, as well as all tests, it does come down to the accuracy of that particular kit and process that the laboratory is using to detect the virus in that sample, okay? And what we are seeing is that you know, the saliva tests that are being FDA authorized at this point have been pretty good in regards to sensitivity over 95% and specificity at 99 to 100%. But the understanding there is that a saliva test does have far fewer viral elements than a nasopharyngeal or oropharyngeal swab. So that means the testing on the back end has to be far more sensitive. The limit of detection, LOD, has to be far lower to be able to call that test positive in that particular saliva sample. But we are seeing the technology play out to do that and to do it well. And in particular, if the accuracy can be validated, then no, you do not need to have 2 negative tests, provided we have the accuracy data of that particular test.
Okay, someone's asking about verifying the sensitivity and specificity of a COVID test for return to work purposes, but you've already recommended that employers review the FDA, EUA website to see which tests are valid and which manufacturers are reporting their specificity and sensitivity. Do you have any other recommendations for verification?
No, it's that straightforward. I mean, the effort must be taken to verify the accuracy. You can't just take, at this point in time, taking the face value of this lab is performing a PCR test does not mean this lab is performing a highly accurate PCR test. And so taking the due diligence to understand and capture that data, and in some cases, the additional step of picking up the phone and calling the lab and asking to speak to the lab manager is appropriate. We do that from the clinical side in terms of vetting these operations, these labs that we partner with, and it's a prudent step for any employer to take that extra effort with the local labs that are provided. If you know in your area there's only 2 or 3 options for COVID testing, then the burden of picking up the phone and calling each one of them to try to validate the accuracy of the test will, at the end of the day, give you less heartburn when tests come back negative and somebody's symptomatic, right? So I'd strongly urge to take the extra step, do the due diligence for capturing that data.
Here's an interesting scenario. How do we assess or monitor testing if our staff are going to free testing sites in parking lots?
Sure. I mean, if you're local, or even if you're not local, right? Just identifying who's doing the test, what vendor is it, and just saying, look, I'm a local employer. I have employees showing up to do testing. I want to get data on the accuracy of your test, and looking to them to provide that back to you is a straightforward answer to that for sure.
Okay, here's one about mutations. I have heard that there have been mutations of the COVID-19 virus. Is this true? There have been reports a person can get the virus twice. Is this because of mutations or lack of immunity?
So we have addressed this in a couple other sessions, so I would encourage you to go more in depth on these 2 particular topics. Can someone get reinfected? The prevailing evidence is still at this point in time, no. We don't know how long immunity lasts, but we do know that there is some imparted immunity once you've had COVID-19 once. Now, that could be for 2 months, 2 years, or 20 years. We just don't know, as we don't know with other illnesses such as tetanus, right? We don't know how long immunity lasts from a booster from tetanus. We think it's 5 years. It may be 10, it may be 8. So the SARS-CoV-2 follows that same clinical logic. Now, with regard to the mutations, yes, there has been documented studies looking at mutations of the spike protein up to 14 times in some cases. And so we will pay attention to that in regards to its effect on creating a effective vaccine, but we don't know to what extent the spike mutations have imparted any increased or decreased virility in regard to the infectiousness of the of SARS-CoV-2 itself.
Okay, we have time for one more, and then we'll answer the rest in our weekly update that we post on the website. There's several questions that have come up lately, and including today, about the intersection between influenza and COVID, and now with flu season coming up again, probably it's going to continue to raise questions about how to handle the upcoming flu season in conjunction with COVID. Maybe we should do that as a topic in one of the coming weeks. But this particular question is, has any study been done on how the extra efforts at containing COVID spread affected the regular flu fatalities in this past season?
Yes, there has been data looking at, another way to get at that question is, what do we normally anticipate in regards to burden of death, mortality in a particular geographic over a particular period of time as compared to now with regard to the onset of COVID in the pandemic. And we've presented this data in regards to excess mortality being observed. We pointed out, I believe, New York having a 2.1, 2.2 increased mortality above and beyond what they normally would experience at this point in time, being obviously flu season approaching. So those data sets are being captured, and those comparisons are being made in regards to excess mortality as a result of COVID-19 in particular, as compared to the flu.
Okay, that concludes our session for today. I just wanted to invite you, if you'd like to submit topics that you'd like us to address in coming weeks, you're welcome to email them to me at karen.o'hara@workcare.com, and I'll share them with Dr. Harris. As always, we really appreciate your participation, all your questions. It's very informative for us, and we appreciate your ability to join us every week. Thanks again, Dr. Harris.
Thank you, and everybody, we'll see you next week. Stay safe.